आज ये कहाँ चले गए तो ये देखना कल का क्या है, कल का सेटिंग।
So, Jijjadu sir, shall we start now, sir, the second session?
Yes, ma'am.
Yeah.
The resource person we have for this session,
Yes,
Mr. Chinmay Sawant, he will join very soon in 2 minutes. And
yes, sir. Yes, sir.
we will
Before
start
that, our international speaker also, our international chief guest also joined, Dr. Dixon Adams, sir.
Yes. Welcome, sir.
Very warm welcome to you, sir, and thank you
Good
so much.
morning. Good morning. Yes.
Thank you so much, sir, for joining with us and sparing your valuable time. Would you please speak a few words, sir, for a while, for a minute?
Yes. I believe you can hear me.
Yes, sir. Yes, sir.
Yeah. So good morning from Ghana, specifically from the Kwame Nkrumah University of Science and Technology. And we are in West Africa. I also send you warm greetings from the INTI International University in Malaysia, where I serve as a research fellow. And I was in a meeting not long ago with staff and students of the Pangasinan State University in Philippines. They all send their warm regards to those present at the conference. It is with profound gratitude and great humility that I stand before you as one of the chief guests of this important gathering to celebrate the World Nature Conservation Day 2026. I'm happy that Sidvi Foundation is organizing this webinar session or this conference to mark this event, this landmark event. Yesterday, I was speaking to Dr. Nakiela, and I realized that she wasn't feeling well. She was even at the hospital, but she was eager, as always, to roll this conference because of her passion for academia broadly and for recognizing efforts of individuals and organizations towards the conservation of nature. I believe that today is not just another day on the global calendar we are using to commemorate our conservation efforts, but is a solemn reminder that the earth, which is our only home, requires that we collectively protect it and then engage in responsible management. At this point, I want us to ponder over the famous remark made by an environmentalist called Baba Diom. He said that in the end, we will conserve only what we love, and we will love only what we understand, and we will understand only what we are taught. So for conservation to begin, it always begins with knowledge. And the knowledge will sow seeds for appreciation, which will then lead to responsible action. So the point I'm trying to drive home is that we have to steer up our sensitization and educational efforts in our respective circles where we work, in our institutions, with communities. And then I believe that when we're able to sow the seeds of knowledge, it is then that we can realize the conservation of nature that we are expecting. We know that nature is the foundation upon which civilization stands. We know the forest provides us with oxygen and all the things that life depends on. Our rivers sustain agricultural activities, and our oceans also regulate our climate. And we know that we need ecological balance. It's important for our survival. That is why the United Nations SDGs 15, which calls upon nations to protect, restore, and promote sustainable use of our ecosystems, as well as SDG 13, which also calls for urgent action to combat climate change and its adverse impacts, are very key today. So as we mark today's event at this conference, I wish all presenters well, and I believe that it will cheer all of us on to help save nature and to help us in always achieving a greener, healthier, and more prosperous world. So I wish all of us well in our efforts to save nature. Thank you.
Thank you so much, sir. Thank you so much for your wonderful words, introductory words regarding this conference and your all-time support to Sidvi Foundation because this is the divine gathering, sir, by means of God. I know how much busy you will be in your own schedules, even though you have joined your busy schedule in spite of your busy schedule also. Thank you so much, sir. Thank you so much for your wonderful speech. Thanks a lot, sir.
You're most welcomed, and I'm happy to always join. And like I mentioned, I really appreciate your efforts in bringing scholars together at landmark events like this, the World Nature Conservation Day. So keep it up, and I wish you the very best. Yeah.
Thank you, sir. Thank you so much, sir. Thank you. And now I wish to hand out to Dr. Sidvi sir for the next session, sir.
Yes, ma'am. So Mr. Chinmay Prakash Sawant, he has also joined. So let's begin with our session 2. For this particular session, we have one distinguished speaker with us, Mr. Chinmay Prakash Sawant, who is wildlife biologist, Wildlife Institute of India. In short, I will introduce him, the work he has done, which is a very splendid work. He is a wildlife naturalist and grassroots conservationist with a strong academic foundation in fisheries and wildlife science. He is experienced in ecological fieldwork, biodiversity documentation, and community stewardship and outreach. He is passionate about bird watching, marine ecosystem, and engaging local communities in nature education and conservation action. As far as his academic background is concerned, he has completed his master's degree in wildlife science from Wildlife Institute of India, Dehradun, and his bachelor's degree is from fisheries from the College of Fisheries, Ratnagiri, which is affiliated to Konkan Krishi Vidyapit, Dapoli, Ratnagiri. As far as his work is concerned, a very uh, large, I should say, the work he has done till the death. He is working as a project associate with Wildlife Institute of India, Dehradun, Uttarakhand, India since 17th June 2026. He is working under the Department of Protected Area Network, Wildlife Management and Conservation Education. The project titled Identifying Effective Wildlife Management Interventions for Replication, which is the best wildlife management practices in India, which is part of Ministry of Environment, Forest and Climate Change, Government of India initiative. His master's thesis, the topic for his master's thesis, that is understanding aggregation sites of elasmo branches in selected islands of the Lakshadweep archipelago. The project aim was to understand the species composition of elasmo branches in selected islands and identify and characterize the known aggregation sites with the help of traditional ecological knowledge, in short, TEK. He has been a leader for one of the project which he has completed very recently from May 2023 to March 2026. He and his team worked on Kiraksal conservation project which was funded by WWF India under Conservation Catalyst Program CCP 2023-24. The project aim was to conduct research, wildlife monitoring, capacity building workshops, and documentation to promote conservation in Kiraksal, which is a very small village in our Man Tehsil, district Satara, Maharashtra state. He has been worked on People's Biodiversity Register, that is PBR, with local BMC in Kiraksal, Satara during March to November 2020. He is having a conservation and protection of grassland ecosystem. He worked on that particular point, as well as biodiversity conservation and community-based awareness in Kiraksal and the allied areas. He has organized biodiversity awareness and educational sessions across villages in Man and Khatau talukas, district Satara, state Maharashtra. He has conducted training camps on wildlife conservation, snake awareness, and human-wildlife conflict mitigation. He has collaborated with Forest Department to expose illegal activities like khair tree smuggling and wildlife poachings, etc. He lead the preparation of People's Biodiversity Register, that is PBR, in partnership with local BMCs, completed between March and November 2020. He worked with locals in conserving the habitat of key grassland species like the Indian wolf, striped hyena, Indian fox, and jackal. He led the Kiraksal Conservation Project under WWF, India's Conservation Catalyst Program, to promote community-driven biodiversity conservation. He led biodiversity documentation and training initiatives in the Kiraksal region. So having such a vast and big biodata, he is also having human wildlife conflict management. He has worked on that. Then wildlife research and monitoring. He has worked on that particular point. So a number of articles in local newspapers as well as in some research papers. He has already published 12 such a type of research publications or book publications have been published by Mr. Chinmay Sawant. And he has also participated in different discussions and conferences at national and international level. As far as his achievements are concerned, he is facilitated as Species and Habitat Warrior Award 2023 by Mumbai Tarun Bharat and Maha MTB under Budding Naturalist category on the occasion of World Environment Day, 5th June 2023. He has been facilitated by Best Alumni of the Year 2022-23 at Junior College Mumbai University, NJ Acharya and DK Marathe College, Chembur, Mumbai in February 2023. Satara Forest Department felicitated for achievement in biodiversity and wildlife conservation work in Kiraksal region, Satara in August 2022, as well as award in recognition of exemplary work in ornithology, that is Udvanmukh Pakshi Nirikshak of the Year by Hope, that is a Nature Trust, Thane and Maharashtra Pakshimitra in January 2022. He is also the member of Maharashtra State Coordinator of Indian Youth Biodiversity Network under the United Nations Convention on Biological Diversity. He is a member of Biodiversity Management Committee, Kiraksal Satara, life member Maharashtra Pakshimitra Maharashtra, and ex-student member Bombay Natural History Society Mumbai. So such a eminent person is here with us in the morning. We have also felicitated him with award, that is Environmentologist 2026. I warmly welcome Mr. Chinmay Sawant in this conference from Sidvi Foundation as well as from our college and our samstha. Welcome to you, Chinmay Sawant.
Thank you, sir. Thank you, sir, for the very brief introduction. I am very grateful to Sidvi Foundation and Dr. Sujit Jadhav, sir, for inviting me to give a talk on wildlife conservation and grassroots initiative, which has been seen in our Satara district. So we will start with our presentation. So I will share the screen. So just tell me, is it visible now?
Yes, yes. The screen is visible now.
Screen is okay.
Yes.
Okay. So, as sir has given a very brief introduction regarding me, so no need of telling more regarding about myself. So, as sir told about my experience, I have worked. Currently working in Wildlife Institute of India, and I did my masters there. And in thesis, I worked in Lakshadweep. And also, apart from that, this is a Kiraksal conservation project which is granted by WWF India. So I have giving some glimpse of this project, like how its initiative has been started in a local grassroots level, and how we can see the conservation with the people, with the communities after we engage them in proper streamline in this field. So this project is under the Kiraksal Conservation Project, Conservation Catalyst Program, which is run by 2 years from May 23 to October 24, and after that this is first phase, and after second phase started from October 24 to March 26. So I will first tell you most of the people here know about the conservation, but wildlife conservation. So here specifically, I am talking about the conservation of wildlife. So conservation, what we see the conservation meaning here, the protection, the management and sustainable use of natural resources in the wild. And also very important, the protecting endangered species and habitat, which is sustainably managing resources that are not be overused. So previously, what we see the conservation perspective, when we see the Indian history, we have seen the conservation starts for the trees. The Britishers start for the timber plantation. They started in the Western Ghats, then in the Central India landscape for the production of timber for the European railways. So from that, it's little bit started of forest conservation. And but regarding the wildlife conservation, the initiative has been started after the 1972, when the Indira Gandhi was a prime minister at that time, and she initiated the Wildlife Protection Act 1972. And at that time, our conservation field makes success. So we started initiated Project Tiger, then Project Elephants, and now we have been working on every taxa, every flora species to be conserved. So the next concept is about the grassroots conservation. So the grassroots conservation, when we talk about that, like for example, like most of the people like working in grassroots, but exactly what they are working on the grassroots level. So the local communities, individuals from that group only, they make a one small group and they started initiated the documentation, protecting their wildlife, protecting their landscape uh, in their community only, in their village only. So here it's not driven by governments, not NGOs, but they can collaborate with the people, local forest, neighborhood, restoring the wetland with the indigenous community. So this is very important thing that grassroots conservation and wildlife conservation definitions we'll able to know. So how it helps? So this grassroots conservation helps in the local knowledge, hence the stronger, longer lasting things. For example, the traditional ecological knowledge, low cost and scalable. It's not like that we have need lot of funds for them to be drive. So whatever initiative at the local level the people is concerned, so they can be taking the initiative and they started this project. And in this, we have started this work in this is our landscape of Mandesh. So here you can see the Satara district, Sangli district, and Solapur district is in the Maharashtra state. And the Man Taluka, which you see in the middle part, is a centralized part of this area. And we have seen this area is a mostly of drought prone area. So when we see this landscape and ecological point of view, it's mostly of grasslands, scrublands, and very less ecological knowledge we don't know regarding him. So that's why it's initiated us to build a strong thing to work under this landscape. So as you see, we initiated the Biodiversity Management Committee. So we initiated Kiraksal Biodiversity Management Committee in March 2020 with the help of 2 to 4 young nature enthusiasts. And we say our lockdown period has helped us a lot to gain this initiative. Because people in the village, they have ideally have sitting there and they don't know like what to do. So farming is also not practicing properly at that time. Water issues are there. And we initiated that this could be a very good initiative to engage the people in this conservation field. And also it's have a scope. So mostly what we say in our area, so that area is a drought prone area. So no need, there is a no wildlife exist. There is a no wildlife other than these small birds or any creatures there. But that is our question to initiate it, that is there species exist in these grasslands, in the scrublands, in the drought prone area? And we started our primary objective with the community to protect the biodiversity of the region and community participation to encouraging local villagers to actively participate in these conservation initiatives. So we initiated night patrolling stuff, then Pakshi Saptah, that is a bird week, and also the monitoring of birds, then butterflies in our village with the help of the local communities. So here I will explain a little bit about the wastelands. Why it's called the wastelands? So this is that hill you are seeing. This is not like you can see, this is like some you can see able to in some Africa area or some savanna, but this is in the our Maharashtra state only, and it's a natural photo. It's no filtered and all. So you can see the vast patches of grasslands, and it's a home to a Indian grey wolf. It's apex predator of grasslands. So the reality, the myth, what considered previously it was wasteland. There is a no trees are growing in this, so it's a wasteland. So Britishers, from Britishers' point of view, it has been a wasteland to all. And so the developmental activities has been initiated in these wastelands. So the reality was, it's not a wasteland, it's an open natural ecosystem. So open natural ecosystems considers of savannas, then scrublands, ravines, and short grasses area landscape habitats that considers our Satara districts around 25.6% of area. And it's quite big because Satara district, you will see in the western part, you have a Western Ghat areas, evergreen forest. It's a high biodiversity hotspot. And in the eastern part, you see this drought area has been a open natural ecosystem. So it's a very good landscape to be understand what exactly this landscape exists. So their feature, it's of grassland only, and it's have a home to different species of wildlife. So what we understand with this, so 18 square kilometer of our Kiraksal village, and we have wolf there, hyenas there, foxes there, and that creates us to be do a little more detailed study on that. So here you can see the study area. This is our India and Maharashtra, and this Satara district is there, and you can see this is the star marking. This is the Kiraksal village, and this is you see Zawali, Patan, Mahabaleshwar. It's having the Western Ghats part, and in the eastern part we have this drought prone area. So it is very diverse district we have. So in the western we have around rainfall around 3000 mm, and the eastern part we have very less around 400 to 500 mm rainfall. So here is a map we made with the community. We sit with the community, we explain them like this is the project we are want to initiate it in our village. And first we made a rough about the villages, rough about the diagram or a schematic map, like how to be proceed to work about the conservation things. So as we say, the Kiraksal village, it's 18 square kilometer, having the 616 hectares of forest. Then surrounding it's around 10 to 12 villages. So from three sides, it's covered with the hilly terrain, and in the other sides is having the riverine patches of Gondole Budruk, that is in the Manganga River, and you'll see that it's vast, very ecologically good thing, but recurring droughts and severe climatic issues has changed the landscape very severe in last 10 to 15 years. Previously, this landscape was very initiative. It was very good to be fertile land also, and also very good for the wild. So this is our study area. So you can see the Kiraksal village lies on the border of Man and previously at Khatau Taluka. So it's on the border of Man Taluka. You can see it's a hilly terrain. We have lot of there, and very good geographical area we have for our village. So our three objective is to be monitoring the grasslands, then documentation and involvement of local in here. So the importance of our project is to be habitat diversity. So diversity here we say about the species richness and relative abundance of the species that exist in our this landscape. So it's have consist of rich ecosystem, grassland habitats that serves a very flagship species of Indian grey wolf and striped hyena. So also this part is comes under the wildlife corridor, and which is a part of integral part of Atpadi Dubai Kuran Conservation Reserve, which is existing in the Sangli district. So our objective of this project is to emphasize on research, then wildlife monitoring, capacity buildings of the youths, of the villagers, workshops, and documentation of this biodiversity, what exists in this Kiraksal to promote the conservation. And we worked on these three principles because we have fixed some targets, we have fixed some things to be achieved in the last. So we achieved these things in the last, but we initiated some formulated some principles to work on that. So wildlife monitoring and research we started, then capacity building workshops about the people, about the villagers, youths, then forest department also, then wildlife documentation, whatever is exist here, we have to be document that. because we don't know exactly what species exist in this area. So that's why whatever we are getting, we are documenting. So we initiated surveys with sign surveys regarding seeing the tracks, markings of the wildlife animals, the den surveys, the night survey we initiated in the night to see the wild animals or any other fauna species, then camera trap installation, then questionnaire survey we did, then wolf data collection. It's a very important thing we did in this project. And we initiated this wildlife exist. So first photo is of um, golden jackal, then striped hyena. It's a den of striped hyena. It's a pug marks of striped hyena. You can see the downwards photo is the Indian fox. It's also called Bengal fox. And it's pellets of porcupines. Then it's a scat of jackal and it's a den of hyena. So you can see this diverse habitat species what we have recorded while our documentation. And this is our very hardworking field. Those who field works, or we can say field assistant, our green guardians, because previously they were doing hunting practices. So we initiated them in this program, and they started working as a monitoring the wildlife, as a started little bit working about the ecotourism activities, and they know the each and every places of the landscape because locals only know every detail of that area. So inclusion of 18 routes, we initiated 18 routes and 2 kilometers area we walked, and total 36.2 square kilometer we surveyed the area. And we also initiated camera trap. And what we found? So we found like Indian grey wolf presence in 13 sites of it's around 68.4%. So this is we did of encounter rate. So encounter rate, that means number of signs per kilometer of animals. So we mark 2 kilometers. So we walk 2 kilometer at every kilometer. How much signs you get? So that is the encounter rate we initiated. And we see it's occupancy based, and we see the occupancy of a striped hyena occurrence. It's around 63%, and mostly dominant is Indian grey. So Indian grey wolf is moving in our whole landscape, then afterwards striped hyena, then Bengal fox and golden jackal. So that four canids, which is also called as a indicator of the grasslands, they are very dominant in our landscape. So we documented around 16 to 17 species of mammals. So in that we found around 10 species of mammals, their signs we recorded. So this is the mammal presence you can see. So this other border, you see that is a forest area. And this is a small streams, then roads, then grasslands is there. And that orange color, purple color mark, that is of mammal den sites. And these wolf locations we have initiated that. And also we find some wolf encounter rate also at every 3 years, because we want to know like how the wolf is moving in this landscape. So we from last 3 years you can see the data also. In 2023, the wolf signs is 26 and distance they travel around 57 kilometer. So at every 0.4 kilometer we have signs. But in 24 also it was good number that we initiated. But in 25 you can see it's very reduced. So it's indicator of that like how the habitat is been changing. So in the reduced in the sense uh, they are not like extinct in that area, but they're moving a lot in different places. So that's why we got a very low encounter rate of that in 25. So again in 26 we are now working, so that data is not yet published, but we are working on that to get is it their encounter is good or not. So as long we have initiated the data, we'll able to get to know like how the species is been moving in this landscape. So here also we have did about den surveys. So we have initiated the mean depth of the den. In the rocky terrain, we see around 2.35 meter depth of measurement of the depth of the rocky terrain. Grasslands we found 2.11, in scrublands 1.96. So in dense scrublands we found 1.70 meter depth. So maximum the dens are in the scrublands, and that is being used by the porcupines and hyenas. So you can see the scrublands, how it's very important for the this grassland species, scrubland species for their survival. So on this data, we will able to get to know on how we can we help this species to be manage their species specific conservation measures that has been initiated by the forest department, by the locals on the base of this data. So this is the den mapping. So in Madladara area, which is in the forest of our Kiraksa, we have a lot of den sites in this. Then in Dambitimbi Hills, that is of hill terrain, 800-meter elevation is there. That also we have den sites there. So you can see that a geographical area, like how the species is been exist in that particular area for they getting a very good microclimate, good food, good cover, and good shelter. So that's why the species been exist in some restricted area only. Also, we did activity pattern. So on the basis of the camera trap, we initiated like when this striped hyena has been active, when this golden jackal has been active. So we have see the striped hyena has been active more in the early morning, 3 o'clock to the from night 9 o'clock or 10 o'clock to the early morning 3 o'clock. It's a peak timing of that. And till it's active in 6 o'clock, because it's a full nocturnal species and it's not seen in the diurnal time. But golden jackal, you see mostly it's a nocturnal only and also the diurnal. But diurnal, they are seeing very less. But the peak activity time is from morning 6 to 9. So that seems this species is very adaptable to this human dominated landscape and can be exist with the humans, and they are avoiding the humans at the night at the morning time because of the livestock pressure. Then people are working, traveling, and that's so they have very coexistence with the local communities. So this is the species we found. So wolves, jackals, foxes, hyenas, small Indian civets, mogos, palm civets, jungle cats. These are we have found in our camera traps. And this also we have seen in our camera traps, wild boars. So this is a very interesting thing we found it. So we initiated camera traps for the mammals and we placed 2 camera traps in front of uh, the 2 trees, and we get 2 photographs in each. So when we initiated this for the hyenas, we'll able to capture this individual ID. You can see at every hyena there has been a different patterns. So like tigers, leopards, we have rosette patterns on their. So the tigers we have stripes. Also the leopard we have rosette patterns. We can identify the individual. On same basis, we can also identify the hyena because they are marked animals. So we can see there. So there are very unique characteristics. So we have identified 5 individual and 2 juveniles, and these 2 juveniles are now become adults. So it's a very interesting to us to document this all. And also at the local level, we have keep them names of the hyenas. So like some names are like on the basis of their behavior, on the basis of their structure, and you can see HRK4 means hyena HBK4. This is photo HBK4. H means hyena, B means Bombay, K Raksal, and 4 number hyena. So this hyena, we given the name Kubda. Kubda means like it's protruding a skull and it's a body. So like this initiative we have given. But we are discussing with this community, discussing with the youths, because how we can give our wildlife names, how we can give our local names to these animals. So the people also feel they are like our wildlife only. That is our things. So they will not disturb or they will not try to harm this wildlife. So you can see this pregnant jackal and can see that Haina is taking the live one. In the morning 7:40, you can see he's going and taking the live goat. And this is the pups to a jackal fox. Jackal pup near the dam. So what we found? So we found 606 species of biodiversity. You can imagine like 606 species in one small village. We have 212 birds, 18 mammals, 160 plants, 34 reptiles and amphibians. And it's a very good data for a village level. So we can't imagine in the one village we have 606 species, but what we have, we surveyed in other villages, 10 villages, what a vast area of biodiversity we able to know. So that's why it's very important to the I also tell to the students who are watching this my presentation, who are watching on the live on the YouTube. I tell to the students, I tell to their professors also, like initiated a small projects in their small villages. Try to learn them to get documented this species, get documented this small things, because that only it's a very important. Neither it's very important to be working in some big landscape or working on some charismatic species like tigers, then elephants. That is also good, fine only, but at the village level, at the local level, what species is there, what wildlife is exist in our neighborhood, we should know that. So I think that will be a very good for them to learn. So you can see this is a great biodiversity of Kyrgyzstan. The first photo, it's of a blue mormon. That is a straight butterfly of Maharashtra. And we have documented in our drought prone area. So it's a good thing about this butterfly, or we can say they are migrating this species in the monsoon time to this eastern part. And for in the western monsoon, we have very large rainfall in the Western Ghats, and that's why they are migrating in the eastern for the relief. And mostly on the lemon tree, citrus trees, they are depleting their eggs. So second photo is of a long-tailed shrike. It's a village bird of Kyrgyzstan. So we have national bird peafowl, right? Peacock. Then we have state bird of Maharashtra, that is yellow-footed. And we have a village bird, that is long-tailed. And third photo is of Russell's Creek. Then can you mute the mic?
Yes, sir, we'll do that, sir. We'll do that. Please go ahead, sir.
Everyone is requested to please mute yourself. The lecture is going on very nicely. So please mute your mics.
I had muted, sir. I had muted them.
Thank you, ma'am. So you can see the third one photo is of Russell's kukri. It's not very common snake we have seen in our grasslands, and it's a very the distribution is a large, but we have very less known about this species. And also the fourth photo is of European roller. So you can say the name on the European, it's come from the European, it's a passage migrant. So what we mean by passage migrants? So passage migrants, they are coming and setting for they are setting for like around 1 or 2 months, and they're leaving to their destination. So these passage migrants, these European rollers, every year they are coming in the month of September, October, and they will stay for Kyrgyzstan, they will stay for this area for some time, for like around 15-20 days in September month. And after that, they leave towards their migration route to other places. So it's not about the resident birds also, it's about the wintering grounds, it's having the passage grounds for the all species. And uh, fifth photo, you see, this is a Siropecha bulbosa. It's a small tuber-like umbrella species, and it's a very indicator of a good vegetation or good floral things in our grasslands. And down you can see the balloon, that is a marble balloon frog. You can see it's uh, very beautiful frog we can see in the monsoon times in our grasslands. And we have termite hill gecko. Then this is jackal, and this is our resident turtle, that is a Indian flapshell turtle. So you can see a very unique diversity of biodiversity in grassland in Kyrgyzstan. And we have to be get to know from this documentation. So like this previously only I told, like apart from that 56 species of these insects also we found. Also then 34 species of reptiles, amphibian, 18 of mammals, 7 species of fishes, 13 of these spiders, and 15 of these dragonflies and damselflies. 91 species of butterflies we have. And it's a very good initiative. We work with the local communities, with the youths to document this wildlife. And apart from Kiraksal, we have very adjoining forest patches. It's in the Kukudwad. It's having a very diverse stock of floral species. So we have 295 species of flora there. Also in that 15% are endemic to that area. So in that Kandil Pushpa, it's also called Ceropegia species, what we see in this. It's a one of a Ceropegia subspecies Lucy. So you can see this species in the month of September, August to September in our hilly terrace. So this is the Kukudwad, Agaswadi, Pukudwadi forest area in the Man Taluka, and we have a very diverse of flora species in here. It's a very interesting sighting we got in the recent only in December 25. Chestnut-headed beetle, also in Marathi we call Badami Dokyacha Raju. So you can see the map. So the violet color dots you can see it's moving, and you can see the distribution of the species in there. And that red color mark, this is the species where we found in India. Where we found in Kyrgyzstan. So this is not their route also to move. This is not their route for their passage migrant also, not for their migration route also. So this is the species how they are came in this. It's a vagrant. It's like they are moving in a group, and some are 1 or 2 species. They followed and they stay for one day. And next day, when I seen for the bird watching, I went for the bird watching. The species is not there. So very interesting sighting I got. So that's thing that it's very important to document. We don't know like how the species can move from one place to another. That's why it's very important to document whatever things we have in our neighborhood. So capacity building workshops we initiated with the local communities, then schools, then we initiated the Wolf Day, International Wolf Day program. So I'm very proudly, I will say, Kiraksal is a village in India which started International Wolf Day in the year 2021. From 2021, we initiated the village level International Wolf Day, and it's the first village in India to celebrate that. And that initiative has captured a lot of public then press also, that a village in India that's initiated this village Wolf Day program, and that changed the perspective of people, those who are working in the wildlife, also those who are working in outside the wildlife. That is a wildlife also exists outside the protected areas, and it's a very important to know the species that we lesser known like wolves. Previously, we hated of the wolves, and it's now a very important to be preserved. If wolves will not preserve, we will lose the grasslands. For example, if we not preserving the forested, so that's why the tigers are lost, tiger will be lost, then elephants will be lost. That same thing has happened in the grasslands also. So we initiated that. Also, we initiated the hyena awareness program with the forest department, with the grassland trust NGO, and we work with them a very lot, with the local people, with the local schools, to initiated that this species, it's a very elusive and it's very little known about. It's a scavenger species, and it's a cleanliness of village, because vultures are not there. Now vultures are taking care of their, means they're replacing the vultures, these hyenas. So that's why it's very important. It's a cleaner of our village, hyena. Then this is a very important thing we initiated in village level, and it's a not a very uh, what we say, it start at some places it can be a very good initiative in early stages, but some places it takes a lot of time. In our village it takes a lot of time, but that initiatives happened a very life, we can say a very good outcome for that. So Kurad Bandi, banning of trees; Shikar Bandi, ban on hunting; and Vanwa Bandi means ban on wildfires. And this initiated village has initiated in the Gram Sabha, and this three sutri we important we initiated village level, and it has been considered a very good practice of biodiversity management in the Kiraksal. So like previously only I have said that these local hunters which we've been for the doing for hunting pressures has been now converted into these local guides, and this is our a very good outcome of this project too, that how these hunters has been transformed to this initiative. So that's why brainwashing of these people regarding not only about telling them this is the good, this is right, but giving them experimental things like how we can be engage them in the proper conservation thing, and how we can be taking this initiative to work towards our betterment of the future, betterment of the livelihood, and betterment of our village. So here we initiated signage boards in different places. So wildlife prone area we initiated. So this youths only with me, youths only has initiated this program, and its impacts a lot. So people, those who have previously been hunting in neighboring villages also, they now stop because of this scared. They scared. वो डर गए थोड़े से ये लाइक किरक्साल विलेज में अभी ये हो रहा है, तो लाइक वो इस पे कार्रवाई करेंगे, उस पे वो फॉरेस्ट डिपार्टमेंट को बुलाएंगे. सो दे इनिशिएटेड दैट दे आर ऑल्सो स्केर्ड अबाउट देम, अँड खूप चांगला उपक्रम गावातल्या लोकांनी हा केला. सो LULC. So LULC, you can see land use land cover. So by seeing the map only you can able to identify, right? In 2020, you see the croplands, which is very less. In 25, we have lot of croplands. Also, the water body is also very good. And we shrink of open savanna. Open savanna, what we called as a wasteland, what we called as a grasslands, they shrink. And due to that happened, uh, this wolf species also disturbs. And also we have seen the tropical thorn forest, which was previously scattered in some places. Some places they makes a very dense, and that makes a dense for the wild boars to come. So from this map, we can able to identify like how the land use can changes a lot in different places. So here I have make a map to understand the village in Marathi only, to get to know like how our landscape is changing and what we are getting from that. You know? So what challenges we have? So we have invasive species challenges. That's a lot thing like antenna camera. Then linear infrastructures like roads, irrigation canals, then power lines, and also we have these windmills, solar panels, stone crushers. It's affect a lot to this. This is a jackal den. You can see this jackal den has been seen in some man-made uh, den. So it's very interesting to they are adapting in some very different species. So here I will I ask you to give some little bit attention to this photo. You can see this is a photo of 2013. At that time, there is a drought area. You can see this is a grassland full of it is. And you can see this is photo of 2019. So little bit of water conservation work has been done. It's good. It's a grassland only. And after the lockdown period, what we see, this is a 2021. We can see the stone crushers has been initiated. And 23 You can see the stone crusher. So see, when we worked in 2019, we started initiated. Our work has been focused on grassland management. In 23, you can see the good number of vegetation also in some places because of our initiatives work has been there. But the stone crushers are affecting a lot. And where the stone crushers are this is a violet color line. It's of a Kiraksal village, and this yellow color line. This is a forest boundary. So it's outside forest boundary. That is a land of this one. What we call private lands. That's a converted into the stone crusher. It's a very devastating thing, and from that it's happened in year 2023 only, March month. You can see the wildfire has been initiated, and 2 hours 30 minutes the wildfire is going on, and around 7 it's going. So around 5 hours the wildfire is moving in this landscape. It's a very devastating to control it, and that creates that we have to be stopped, or we have to be initiated these upcoming things to be stopped. So conservation plan we made, biodiversity monitoring, local perspective interventions, and threats to wildlife, what we have threat. And community participation is very important. Then when we engage the local citizens and local villages, school students, then only the real conservation is happening. So we make action plan. So we have made a 5 years action plan about the strengthening the wildlife research, monitoring, controlling of hunting, habitat destruction, then youth engagement, conservation of water resources. There are around 9 to 10 points, and we implemented with the Biodiversity Management Committee of Kiraksal. We called Sukshma Kruti Arakhada. It's like a micro action plan. And this micro action plan, we initiated with the Gram Panchayat, we initiated with this local body to say that if we are not doing this, so we are facing a lot of stuff in our village. So that's why we are working on that. So this micro action plan tells about the Adarsh Gaon ideal village, then youth involvement, Paramparik Gyan means traditional knowledge, and Jalastota means water bodies conservation. And in first phase in December 24, we met principal associate principal chief conservator of forest wildlife, Dr. B. Clement Ben. He is now IGF, Inspector General of Forest in Delhi now. And we given him the project, and we talked with him regarding some management practices in Kiraksal village. And a historic milestone for this our nature conservation thing. We had in February 26, we initiated a release of report Kiraksal Conservation Project. So whatever I explain in this presentation, it's in the report. So you can also take a screenshot of it, or you can take a photo, because there is a QR code, and you will able to get to read the report in English in detail, like biodiversity assessment of flora and fauna with focus of Indian Caribou habitat. And it's a very detailed report of this, what our work has been initiated and how the green guidance has been followed. So you can see this second photo. He is a Somnath Katkar from our Kiraksal village. He's previously engaged in a very large number of hunting practices. But after we initiated this program, he has been converted this hunting to monitor the wildlife. And we facilitated him in our program to get from the DCF Satara, that's the Deputy Conservation of Forest Satara is there. And we have a chief guest, our Maharashtra's ex-PCCF, Sunil Limaya sir. And this has been get to know to the people that they are like, it's a very good thing to be conserved, because ये हमारा ही गांव है, ये हमारा ही विलेज है, इसको हमें बचाना है. So like they have some initiative to do that. So also we give the report to our DCF Satara, District Forest Officer Satara, and also they initiative that
Kiraksal संवर्धन प्रकल्पाचा जो
conservation
अभ्यासावर प्रकाशित झालाय, जो 2023 ते 2025 या कालावधीमध्ये चिन्मय सावंत आणि त्यांच्या टीमने या ठिकाणी अभ्यास केला आणि हा प्रकल्प राबवला. तर या अनुषंगानं वन विभाग यापुढे सुद्धा चांगलं योगदान देईल आणि हा कन्झर्वेशन रिझर्व्ह घोषित होण्याच्या दृष्टीने प्रयत्न करेल असं माझं मत आहे आणि निश्चितपणे प्रयत्न केले जातील जेणेकरून आज माळरान संवर्धन हा विषय खूपच दुर्मिळ होत चाललेला आहे आणि या ठिकाणी जे लांडग्यांचं बोलल्या असा प्रयत्न निश्चितच वन विभागाच्या माध्यमातून आणि किरकसाल ग्रामस्थांच्या लोकसहभागातून केला जाईल. त्यासाठी चिन्मय सावंत आणि बाकीचे जे या ठिकाणचे संशोधक अभ्यासक आहेत त्यांची सुद्धा मदत घेतली जाईल आणि निश्चितच एक चांगलं काम या ठिकाणी उभं केलं जाईल असं मी या ठिकाणी निश्चित सांगेन.
So this is been initiated with the department, and department also has taken a lot of good things to be initiated in this work. So today guy means what is upcoming thing to be done? This much we have documented, we have report, we have a good means of documentation. But what we can do next? So we have some options of community reserve, conservation reserve, biodiversity heritage site, and other effective area based conservation and key biodiversity areas. So we worked on that and we able to know that this is a strategic tri-junction. So previously we have two conservation reserve, Maini bird conservation and Atpadi wolf. And after that we have Kukudwad forest, which is having the floral endemism. And from that three tri-junctions, we have Kiraksala very in the middle part. So that's why we want to see, this is a very important thing to be ecological point of view to conserve. Otherwise, we will lost the isolated and it will be rupture the wildlife corridor of the species. So we recommended the forest department to be conservation reserve to this area because the land is of the forest department and we will initiate this conservation reserve. And this is the map we have seen. So there are three villages map: Bombadi, Dambiwadi, Kaiwadi, and forest area. And this is the area we proposed we are going to be doing. So here we can see the wolves movement in these three: Landagdara, then Valdara, Tilbi Woods. And we want to be save this wolf movement to be kept as it is. So like this, if the conservation reserve not happened, we will see for the biodiversity site also, if it's not happening. And you can see this is the big landscape level approach. The red color is of Khatao Taluka Tehsil, and yellow color is of Man Taluka, and this peach color, it's of Atpadi Taluka, is in the Sangli district. So you can see the village Kiraksal is in the pink color polygon in the middle part, and it's having the vast of grasslands patches in with it. So you can see these grasslands patches here, and you can see the Maini Bird Conservation Reserve, what we're talking here. Here Atpadi Wolf Conservation Reserve, and in that Kukodwad, it's a tri-junction, and in that Kiraksal is here. So you can see how we can able to see this corridor conservation. If its corridor is conserved, it will able to help to this species, help to the wolves to move around from here to there. So you can see this is a red spot is a wolf movement, and this is the grassland patches. And here you can see the wolf movement here. This is the wolves we recorded from Varkote Malawi. And what we exist. So here you can see the Maswad College is here near to Varkote Malawi only, and you can see this is all grassland patches there. And the red color, the polygon inside in that is the solar panels. Solar panels are proliferating in these grasslands, and they are thinking like they're getting solar energy. Solar energy are getting to whom? They are getting some outsiders only, not the local people. They are getting any solar energy. So they are devastating this climate a lot. And what the important thing we are getting? So here grazing rights are very important because mostly Dhangar communities, shepherds are there important in our Mandesh. And coexistence is very important thing. So we are working on these grazing rights and coexistence in upcoming our years. So these two documentaries has been featured. So first is of Kiraksal, it's Species and Habitat Awareness Program by the Habitat Trust and Mumbai Tarun Bharat, which is featured in 2023, and it's available on YouTube also. You can also take a screenshot of this. And this one recent, the Kiraksal return of wolf was been initiated by Green Hub Central India and with the help of WWF India. So this documentary is not yet publicly available now, but in upcoming month it will be available on the YouTube. And I also share the links to the Dr. Sujit sir, so they can also share to you, or you can go through on the YouTube, or you can search the Kiraksal documentary or Kiraksal wolf documentary, so you'll be able to get to know that. So in that, you will get to know that how the village has been initiated these practices in some 7 to 8 minutes or 20 to 25 minutes. So we make this small pamphlets, small infographics to the people to explain them like how our this land use changes. So Kuran, so grassland with forest area, what we say that in the local we call Kuran. So Kuran, how it's declining in day by days. So what we need to be do, and we make a plan. So we need to be do has a biodiversity heritage site, then dense conservation, then compensation will be given to the forest department, and like this we have initiated. And this is the overall thing. So core objective will get to know struggles and challenges. I will say we have a very lot of local political and social barriers. Because when we say about conservation here, firstly, people say, हाँ conservation में तो राजनीति नहीं आती है, conservation में तो राजकारण नहीं आता है. But this is the field having a lot of politics things. Because people will be adjoining you a lot thing in various things. In that barriers are also there. Then we have different types of scientific hurdles, the resource limitations. We have because when we have very limited fund for them. It's around 3 to 4 lakhs, and for the year, and it's a very minimal to get to know explain about the detailed wildlife here. And habitat species threats also very important. So master strategy, what we plan to strengthen this scientific documentation by completing BBR, then social, navigate them from social political landscape, and to leverage them into legal things like policy framework thing, and to be status something regarding to be that can be help a long term benefit, and to build sustainable capacity. Then ultimate goal is to be a legal formation of recognition of VHS or conservation reserve, and it has been a sustainable model for community-led financial viable, a recognized Mandesh region for grassland studies. And that helps a try of Gram Panchayat, Forest Department and WWF Worldwide Fund for Nature. It's a field partnership is very important. And this is our hardworking team who has been successfully implemented this a very without it, this is initiative is not working at all. So this is our all supporter in the sense just the WWF is given the grant, but not others. Other has given some about the technical stuff or some equipment stuffs regarding that, and they helped us a lot in this initiative. And in future thing, we will work on the wolves in this Deccan Plateau, because this Deccan Plateau is very important for the survival. And you can take this my contact details. You can follow on our Facebook, Instagram, and my LinkedIn profile is there. And you can also take a screenshot of it. And this is our small hyena pup. So Kiraksal is a historical thing in Andesh area and for the grassland conservation. It's a very giving a new initiative for the conservation of grasslands, open natural ecosystem in India. So I thankful to all. I thankful to Sidvi Foundation for giving me the opportunity to present our work in this international conference. I also thank to Dr. Sujit sir for giving us time for giving. So sir has called me last week and tell that there is our conference, you will able to give your time. So I told sir I will give the time, because people, the students, the professors, any learners, they should to know like this is the thing they can also initiate it. So it's not like that just the Chinmay is going to be started this work and they initiated, but they also can do initiated in their work also. So thank you so much to all the organizers for organizing this thing. Thank you.
थर्डच्या मार्चला कधीचं पण
Thank you very much, Chinmay, for your nice and very informative
Thank you.
work, what you have shared with us. And I believe that whatever the work you have done in Man Tahsil, which is very guiding to all the society, to the state, to the maybe to the nation, and very soon we will go with some other projects with you.
Yes, sure, sir.
Thank you very much.
Yes.
Now,
So, is there any
Thank you.
questions? So we
Yes,
can
yes.
take the questions.
Not question, sir. Thank you so much. I must say
Okay.
my heartfelt thanks to Mr. Chinmay, sir, on behalf
Thank
of
you,
Sidvi
thank you.
Foundation. And only request, sir, if you could able to give a 1-page note or 2 pages note of your talk,
Okay.
we'll be including in the conference proceedings, sir.
Okay, sure.
This is what I required from your side.
Okay.
Except this, yours is a wonderful talk, sir. Wonderful work also you are doing, and keep going, sir. And I wish many more fruitful talks will be in future. We are expecting from you in our upcoming events also, sir.
Sure.
And thank you, Dr. Sujit, sir, for introducing a wonderful personality to the auspicious gathering. Thank you so much, sir. Thank you.
now I request Mr. More sir to introduce our today's chairperson, Mr. Maruti Petkar.
Sir, sorry, sir, Sujit Jadhav, sir. Before this, we wish to announce the award once again because the morning session, sir, is not available with us. So,
Okay, sure.
if you allow me, I'll announce the award once again, sir.
Yes, please, sure.
I request Mr. Rashekar, sir, please share your screen and display sir's award, sir. Mr. Chinmay Sawant, sir, it's our privilege and honor to announce Best Environmentalist Award 2026 for your wonderful work towards the society and towards the nation and towards environmental sustainability, nature conservation. You deserve it, sir. That's why I had immediately followed up whenever Sujit Jadhav sir nominated you. Our jury members had decided to announce this award to you, sir. Thank you so much and congratulations. And this is your beautiful memento, sir.
Thank you.
Yes. Thanks a lot, sir. Thank you so much.
Very hearty congratulations, Chinmay sir, on this
Thank you,
achievement.
Thank you so much.
Yes, sir. Sujit Jadhav, sir, now you can go ahead for the next sessions.
Okay. So now the next technical session number 2, we will start immediately. For this particular session, we have chairperson, Mr. Maruti Petkar sir, who is assistant professor at Baburao Patil College of Arts and Science, Angar, Taluka Mohor, District Solapur. I request Professor Mr. More, PM, to introduce today's our chairperson, Mr. Maruti Petkar sir.
Thank you, sir. In the second technical session of our conference, we have with us eminent personality, Mr. Maruti Ramesh Petkar, as an chairperson. Now I am going to introduce himself. Mr. Maruti Ramesh Petkar sir is a well-known zoologist. He qualified NET with GRF and pursuing his PhD in Solapur University. Currently, he is working as assistant professor in Baburao Patil College of Arts and Science in the Angar, Taluka Mohor district Solapur. He has 9 years of total UG teacher experience. He published 12 research paper and 1 book chapter. He successfully completed 1 minor research project of Solapur University. He actively attended more than 15 national, international conferences, webinars, seminars, workshop, and FTP. Thank you, sir.
Madam, let's start with our presentations. Mic over
Yes,
to
sir.
you.
Yes, sir. Yes, sir. For this session, thank you so much, More sir, for introduction of chairperson. And I'm inviting the chairperson, Mr. Maruti Petakar, sir, and also the co-chairperson, Dr. Gandhi, and sir. And the first presentation for this session 2, bearing the number ICMIRPSD 2026-07007, kojic acid from microbial metabolism to industrial applications by Mr. Bharat Nirav Sharf or Darshan Masjid, research scholar, Department of Biotechnology, SRKI, Surat, Gujarat. Is Sharaf sir available?
Yes, ma'am.
Yeah, ma'am. Yes, sir. Are you able to present now, sir?
Yes, ma'am. Can I share my screen?
Yeah, please go ahead.
Is my screen visible?
Yes, sir. It is visible, sir. It is visible.
Okay, so can I start?
Yeah, please.
Good afternoon, respected chairperson, distinguished speakers, faculty members, researchers, and fellow participants. Today I'm going to present my topic entitled as kojic acid from microbial metabolism to industrial applications. Myself, Parth Shroff, a research scholar from the Department of Biotechnology at Sri Ramakrishna Institute of Computer Education and Applied Science under Sarvajanik University, Surat, and currently I'm working under my supervisor named as Dr. Darshan Marjadi. I will be explaining how microorganism will be producing kojic acid, the metabolic pathways, fermentation technologies, its applications, and ultimately how this will be contributing towards the sustainable development. So this is my presentation outline. So first of all, what is kojic acid? It is generally a fungal secondary metabolite, chemically also known as 5-hydroxy-2-hydroxymethyl-4-H pyranephoron. It is mainly produced from the species known as Aspergillus, while some Penicillium and Acetobacter species are also producing kojic acid. It is synthesized during the late exponential and stationary phases of the microbial growth. It is soluble in water, it is heat stable, and it is an excellent metal chelator. And because of this property, it is considered as one of the most valuable natural compound which is used in the biotechnology area today. The chemical formula of kojic acid is C6H6O4 and the molecular weight is 142.11 gram. So kojic acid, if we know about the history of kojic acid began in Japan during the studies of koji fermentation, where steam rice was fermented using Aspergillus oryza. And this is also the origin from where kojic acid came into the picture. Initially, it was regarded as simple byproduct of fungal metabolism, but currently it is widely investigated not only as an product, but also it's applicable in pharmaceutical, food agricultural, and biotechnological applications. Why kojic acid is important for us? Apart from the cosmetics application, it is having role in pharmaceutical industry, food industry, agricultural areas, nanotechnology, and in biotechnology area as well. Major producers of kojic acid are Aspergillus species. Among them, one of the most safest organism, which is also considered as GRAS, that is Aspergillus oryza, because the production efficiency from this species is considered as one of the most highest out of all the Aspergillus species. And apart from that, some Penicillium and Acetobacter species are also producing the same. What should be an characteristics for the ideal industrial producer? So organisms should produce high kojic acid. It should be considered as non-toxigenic and safe. It should have rapid growth rate. It should be genetically stable. Also, the substrate should be inexpensive and the scale up should be also done easily. So there are some primary and secondary metabolism which are essential. If we talk about primary metabolism includes those substrates which are essential for growth and reproductive process, which involves cycles such as glycolysis, TCA cycle, pentose phosphate pathway, and amino acid biosynthesis. So when the nutrients will become in the limited condition, this growth slows down and metabolism shifts towards the secondary metabolite. And during this phase, microorganisms will be producing specialized compounds, which is called kojic acid. And apart from kojic acid, penicillin, citric acid, and lovastatin will also be produced. So this is the general flow chart where from the glucose, it had been, we can say, transferred towards the primary metabolism. And if the nutrient limitation condition is arise, it shifts towards the secondary metabolism and kojic acid is produced. So the biosynthetic pathway, which is pentose phosphate pathway, which is one of the most important pathway for the kojic acid production. Unlike glycolysis, which only produces ATP, this pentose phosphate pathway will be producing NADPH as well as ribose 5-phosphate. NADPH will be reducing the power required for the biosynthetic reaction, and this will be helping the fungal cell to maintain its redox balance and also to protect against any kind of oxidative stressful conditions. And for this particular situation, microorganism which is Aspergillus species will be producing kojic acid. So for the production of kojic acid, there should be greater NADPH availability, higher carbon proportion, which is very essential for the kojic acid. This is the biosynthetic pathway, which is basically producing the kojic acid, in which glucose gets converted into glucose 6-phosphate, which enters into pentose phosphate pathway. And after some phosphorylated intermediates, kojic acid is being produced. These are the regulatory genes which are involved for the kojic acid production. The first gene is COJ-A gene, which mainly encodes oxidoreductase enzyme. COJ-R gene, which is important for the transcription, and COJ-T gene, which is a transporter which exports kojic acid from the fungal cell towards the surrounding medium. So the fermentation technologies which is involved for the kojic acid production is submerged fermentation and solid state fermentation. And I have shown some of the advantages of SMS and SSF in the given table. This is the complete fermentation process of kojic acid production, which shows the workflow. First of all, microorganism is being grown. It is then producing seed culture. Media is being prepared. Fermentation conditions is performed. After that, filtration is being performed. Then extraction, purification, crystallization. At last, kojic acid is being produced. Some of the factors which affects the kojic acid productions are carbon source, nitrogen source, carbon to nitrogen ratio. Apart from this, there are some physical factors, biological factors, and environmental factors which also affects the kojic acid production. So the agro-industrial byproducts which is responsible for producing kojic acid in a sustainable way are maize bran, sugarcane baskets, molasses, fruit peels, wheat bran, and rice bran. So this is the workflow of sustainable production where agro-industrial waste is being selected, pre-treatment is being performed, after that solid state fermentation is done, and at last kojic acid being produced. It will be then extracted, purified, and then it will be considered as in useful form. The downstream processing and purification of kojic acid is also being involved, where some of the basic steps which need to follow. The first major stage is biomass separation, where kojic acid the first step is biomass separation, which removes fungal mycelium and it insoluble particles using filtration or centrifugation. Then clarification being is performed. Later on, extraction is being done. After that, it will be purified, crystallized, drying, and packaging is being done. This is again the workflow chart of downstream processing. So how we will be characterizing the kojic acid? It will be done via UV visible spectroscopy, high performance liquid chromatography, Fourier transform infrared spectroscopy, nuclear magnetic resonance, and LCMS or GCMS spectroscopy. Is my slide changing?
Yes, sir. Yes, sir. Please go ahead. Yes, sir. Hello. Hello.
Sorry, due to technical error, I can't see my screen. Just can you give me 1 minute?
Yes, yes, please. Now it is visible, sir. Yours.
Okay, so is it changing?
Yes, yes.
Yeah, okay. So can I begin?
Yes, please.
Sorry for the interruption. So we were discussing the characterization analytical techniques. Later on, the industrial applications of kojic acid are in food industry, pharmaceutical industry, cosmetic industry, environmental remediation, nanotechnology, agricultural areas, and biomedical coatings. So what are the current challenges for the production of kojic acid? Low productivity of wild strain, product inhibition, long fermentation duration, and limited carbon conservation efficiency. Apart from this, what are the process challenges? High downstream processing cost, contamination risk, foam formation, scale up difficulties, oxygen transfer limitations. And what are the regulatory challenges? Product purity requirements, quality control, safety regulations, and cosmetic concentration limits. So, what can be the future research directions for the kojic acid? We can improve the strain with the help of ALE or else CRISPR-Cas genome editing, metabolic engineering, yeah, synthetic biology. We can also do the sustainable production by utilizing agro-industrial waste. We can also promote circular bioeconomy, green extraction technologies, zero waste biorefineries. And in terms of digital biotechnology, we can include artificial intelligence, machine learning, digital bioprocess control, and smart fermentation system. So, how kojic acid is responsible for the sustainable development? We can utilize the waste, we can promote circular bioeconomy, and this all will be ultimately benefiting the environment. Apart from that, we can include sustainable bioprocess engineering. It will also be helpful for the economic sustainability, and ultimately, this will be coming under the sustainable biotechnology. So, how this particular project will be contributing towards the sustainable development? So, here are some United Nations Sustainable Development Goals, in which it is including SDG 3, which is for good health and well being, SDG 9 for industry, innovation, and infrastructure, SDG 12, which is responsible for consumption and production, SDG 13, which is controlling the climatic situation, and SDG 15, which is considering for the life present, yeah, living on terrestrial areas. And I would like to conclude my topic with some of the essential features. Those are kojic acid is one of the most important fungal secondary metabolite, which is mainly produced from the Aspergillus species. Pentose phosphate pathway plays a central role by supplying NADPH and metabolic intermediates. Efficient production depends on suitable microorganisms, optimizing fermentation conditions, and applying effective downstream processing. Use of agro-industrial byproducts makes the production process more sustainable by reducing waste, lowering the cost, and supporting the circular bioeconomy. Future advances in adaptive laboratory evolution, metabolic engineering, CRISPR, artificial intelligence, and green downstream technologies are expected, which will be transforming microbial production into more efficient and sustainable production process. I would like to conclude with my final thought, that is biotechnology is not only for producing valuable compound, it is also producing them sustainably for the benefit of society and the environment. Thank you.
Thank you, Mr. Sharaf. I request chair and co-chair if any questions from your side, sir. I request Mr. Marti, sir, any questions to the presenter? Sujit Yadav, sir?
No, maybe there are no any question.
Shall I go to next presentation, sir?
Yes, please.
Thank you, sir. Thank you so much. With the permission of Honorable Chair and Co-Chair, I am going to next presentation bearing the number 008 under the title Smart Solutions for Skin Health, Evolving Assessment System in Dermatology by Mr. Abhijit Sanjay Velinkar, Associate Professor of PR, Kote Patil College of Pharmacy, Amravati, Maharashtra. Mr. Abhijit, sir, are you there, sir, in the meeting?
Yes, madam. I have already joined. Just sharing my screen, madam.
Yes, sir. Yes, sir. Please go ahead.
Just give me 1 minute. Okay. Okay. Just give me. Madam, my presentation is visible, na?
Yes, sir. It is visible, sir.
Okay. So good afternoon to all participants and judges. So myself, Abhijit Sanjay Velinkar. I'm currently working as an associate professor, PR Kote Patil College of Pharmacy, Amravati, Maharashtra. And the title which I have chosen for the presentation on the topic, Smart Solution for Skin Health, Evolving Assessment Systems in Dermatology. Regarding the abstract which I'm trying to deliver the article, in this article I'm basically focusing on the smart systems which are now available for the mainly I'm focusing on the diagnosis of these dermatological diseases, because we already knew, or it is present in the literature also, that many of these dermatological diseases have much more similarity in their symptoms. So by the casual visual observation, many a times what happens that the physician, due to such a similarity in the symptoms, he wrongly interprets a disease, so wrong treatment can be given, and ultimately the patient could not get any effect or could suffer from some kind of another side effect. So disease diagnosis is very difficult in such a scenario. So I am trying to focusing on the smart system, or you can say the modern technologies, digital technologies, which can help us to eradicate this problem. Also I'm focusing in this article also on the history of dermatological diseases, how they are going to diagnose, what was the treatment was available, then how the advancement has been done in the diagnosis part, in the treatment part, and then I will later come on to the modern day technologies which are available for diagnosis and which are available for the treatment also. So some of the keywords which I have already given here: dermatology, AI, dermascope, teledermatology. See, dermatology deals with the diseases related to skin, hair, nails, and mucous membrane. More than 4,000 skin diseases have been reported, okay, worldwide. And the outpatients which are visiting to the hospitals and to the general physicians, about 50 to 30% cases worldwide are reported to be of the skin disorder. Dermatology in current day has evolved from a casual visual inspection or observation to the AI assisted diagnosis. Some smart technologies are also there for diagnosis, monitoring of the disease, treatment of the disease. All I have covered here in this topic. Critical functions. So if importance of dermatology has been considered, like enable the early identification of skin disorder, assist in the accurate diagnosis of the diseases like skin cancer, psoriasis, eczema. So these are the two diseases, psoriasis, eczema, which have a very much similarity of their symptoms through the casual observation, and many a times physician wrongly diagnose and might lead to medication error also. Autoimmune diseases, cosmetic skin conditions. What might be the outcomes of this dermatology? Like it improves the quality of life, it encourages the prompt medical intervention, of the disease, enables the personalized patient care. That is very important. Okay, so if we are able to rightly identify the disease, we can focus maximum on enabling the personalized patient care with the full impact of the precision medicine. Okay, strengthens the preventive healthcare. That is main important aspect. So we already said the prevention is always better than cure also, and supports a long-term clinical outcome. That is a very important part. It supports a long-term clinical outcome. Okay, on better also. If I say I'm already told that I already told that I will go through the history regarding it also. So here say this ancient Egypt 1550 BC, Ebers and Edwin, okay, Smith Pipery. These are the books which were available in the early ancient Egypt history, where the information about these dermatological diseases, their signs and symptoms, their treatments, herbal remedies, life cycle modification, what should be done. Even ammi juice, it is a seed or it is a juice of a seed, where a first reference to the phototherapy has been found out in some kind of a historical book. Means the phototherapy means you have to apply that juice on your wound and expose it to the to the sun. What happens because of the sun? That juice leads to the formation of some kind of active molecules, or you can say medicinal molecules who can cure that disease. In ancient India, Charaka Samhita, Sushruta Samhita have actually given the classification of these dermatological diseases, their signs and symptoms, their treatments, lifestyle modification, herbal remedies, even the dermatosurgery, surgical procedures reference have also been there. In Greece, the Hippocrates, who is we consider as the father of medicine, he says that you firstly observe the patient and then go for the diagnosis and then go for the treatment. He has given the classification of the skin diseases. Okay, 18th to 19th century, Robert William, he has also given a systematic classification of the skin diseases, and he firstly introduced an entire different branch of the medicinal science, which is the dermatology. Earlier days, a general physician is used to tackle these dermatological diseases cases, but nowadays the separate branch is there. Specialized doctors are there who can tackle these cases, dermatological cases. In 20th century, when we came, microscopy, histopathology, use of antibiotics, use of lasers, use of phototherapy is going to be recommended for the treatment of diagnosis and treatment of these dermatological diseases. In 21st century, recently, AI, teledermatology, dermoscopy, optical coherence tomography, okay, reflectance confocal microscopy, personalized medication are some of the popular names are there for the diagnosis and treatment of this dermatological disease. So key milestones which have been achieved from the very old history to till now, 21st century, is that ancient civilizations have established the descriptions of these skin diseases and the herbal treatments necessary. Ayurveda has also given the classification, treatments, and surgical approaches. Hippocrates have firstly bring the classification of these dermatological diseases with the clinical reasoning, means signs and symptoms related to each and every disease. Robert William led to the foundation of a different branch of a medical science, which is a medical dermatology through this such a disease classification. And nowadays, the modern day dermatology integrates the artificial intelligence, machine learning, precision medicine concept for the right diagnosis and right treatment. So major factors which are considered to be responsible for the skin diseases includes the infections, genetics, immune disorders, environmental, and life cycles. Like some examples I have given here, like bacteria, fungi, viruses, exposure to irradiation, pollution, stress, diet, family history, all contributes to these skin diseases. Now, where I'm saying the smart technology. Smart technology means the technology who have base of CNN, neural network, AI, machine learning, who have those ability to analyze the target and give some kind of a response which is already related to the diagnosis and treatment. Dermoscopy, it is a recent tool which is available. This pigmented lesion, RCM means reflectance confocal microscopy, it is used for cellular imaging. OCT means optical coherence tomography, it is used for cross-sectional imaging. Means when we take up some what we can say, the histological or histopathological diagnosis. Wood lamp for pigment and fungal diagnosis. Skin analyzer for hydration, pores, and pigmentation. use of AI in dermatology. This is very important, and it is now a base part of this smart solutions which are provided. So what is a basic typical protocol which has to be followed is the first you have to take a skin image. I also mentioned here on this image also. Then that image is subjected to some kind of a processing. Deep learning might be through CNN, means neural network, machine learning, AI, deep learning, and then there is some kind of a disease prediction and treatment suggestion are there. So nowadays many kind of this such kind of apps, kind of devices embedded with such apps are available. Okay, benefits, if we say it is a faster diagnosis, higher consistency of the operation and results, reduction of the humans, and remote assessment. Remote assessment means a patient, by using his simple mobile phone and the camera, can took up the picture of the supported skin infection, and that the particular data has been transported to cloud, where a particular dermatologist can analyze it with the help of AI, CNN, and machine learning, and can give a proper diagnosis and proper treatment, which is I have covered in the next slide. Next, we came to the part which is called as a teledermatology. Excuse me, madam, can you close the mic because it create an echo here.
Yeah, I request Parath
Okay.
Sharaf, sir.
Next part comes is the teledermatology.
Yeah, please, sir. Please, Abhijit, sir.
Okay. Shall I continue, madam?
Yeah, yes, please, sir.
Okay. So next part comes the teledermatology. Okay, teledermatology is my next smart solution, which has been, and all these solutions are now been available also. Like the patient, suppose he's suffering from some kind of a skin infection, he can use his mobile app. He could take a picture of that skin infection. That picture will immediately be submitted to a cloud database. Okay, dermatologist will be sitting over there. On the other hand, he analyzed that through image analysis. He could use AI, he could use machine learning, he could use deep learning, he could use neural network for the analysis of that image, and ultimately that one dermatologist could give a right diagnosis, even if there is a similarity of the symptoms, and a right treatment which could be followed. Even through such a app, the continuous monitoring of the patient can also be happen. Okay, many times it is complained that patient do not follow the medication adherence. He take a medicine, as soon as he get a relief from the symptoms, he stop the medication. So such kind of a teledermatology can helpful to the medication adherence also. Advantages of such an approach is the rural healthcare. In rural, the many patients have been suffering from such a dermatological conditions, and they are not easily accessible to the cities where where the specialists are there. So in such a case, that particular patient could use such kind of teledermatology to get a easy and quick healthcare access. Lower cost is also there. Okay, because such apps are available are either of free subscription or could use less amount of money to provide a subscription. Better accessibility is also there. Fast consultation, it is one of the important ones. Because if a particular patient go to a doctor and it is purely crowded with the another patients, so he might have face more time delay to get a right treatment. There are many types of that, like store and forward. Data could be stored on a and forward to the dermatologist, and then he could draft some kind of a conclusion and forward to the patient. Real-time consultation is also possible. Hybrid model could also possible for both of these approaches. Next, modern therapeutic challenge technologies which could be are available, like laser therapy is available, as we already know this. Phototherapy, microbiome therapy, personalized medication are available nowadays for the treatment of dermatological diseases. Laser therapy, laser resurfacing, pigmented lesion, acne scars can be treatment. Skin microbiome resortation with the help of probiotics is also one of the preferred approach. Okay. See, some of the common diseases and their smart diagnosis, how can it be done? Like acne, AI imaging can also be done. Psoriasis, digital scoring. Digital scoring means what? That it is a part of the teledermatology. You have to take up a photo of that particular your suppose skin infection. You have that photo will be submitted to the cloud. In the cloud, that photo right image analysis will be done through AI, machine learning, or neural network, and though the dermatologist who is present on the other side, he applies his individual score. on that particular symptoms. And with the help of that digital score, we can able to know what is the severity of the diseases, what is the disease progression, and what correct treatment could be followed. Dermoscopy associated with the AI, vitiligo, fungal infection with the help of UV imaging, all such a smart diagnosis treatments are right now available also in India, worldwide, and freely are used. So future trends, what might be? AI, big data means data cloud, wearables. So wearables, the smart wearable watches or some kind of any other kind of a device, if support a particular person is suffering from the infection of UV radiation from sun ray, he could use that such a wearable so that he could get right or real-time information about how much he has exposed. Okay, smartphone apps, which is connected with the teledermatology, cloud monitoring, precision dermatology. Precision dermatology means a patient's own genetics, own family history data has been incorporated to get accurate, personalized treatment for this. Okay, so this is how the conclusion which I have drawn, that the dermatology has been evolved from the ancient observational practices to the technological advanced evidence-based medical sciences, and the integration of such a smart solutions, AI, advanced imaging system, teledermatology, personalized diagnostic tool has transformed this assessment and management of skin health solutions. So these innovations not only enhance the diagnostic precision, but also they are preventive, patient-centered, and accessible dermatological care they provide. Okay. Also, digital technologies are continue to advance. Okay, and stands to the forefront of the medical innovation. The future of dermatology always lies in the continuous collaboration between clinicians, research, and technologists to ensure safe, ethical, and equitable access to smart skin health solutions worldwide. Okay. These are my references which I have taken from this data, and thank you. So any questions, madam? Anybody wants to ask some question?
Thank you, Mr. Abhijit sir, for your wonderful and elaborate presentation. I request chair and co-chair if any questions from your end, please. Okay, sir. Thank you, Mr. Abhijit sir, for your presentation.
Thank you, madam. Thank you, madam. Okay, thank you.
With the permission of Honorable Chairperson, I am moving to the next presentation bearing number 009 under the title Green Hydrogen Economy Opportunities for Sustainable Development by Dr. Susmita Srivastava, Assistant Professor, Department of Applied Sciences and Humanities, Institute of Engineering and Technology, Lucknow. Is Susmita ma'am available? Okay. Without wasting much time, we can move to the next presentation bearing the number 010 under the title Screening of Water Samples for the Presence of Energetic and Pyogenic Bacteria in Swimming Pools by V. Pushpalata. Pushpalata ma'am, are you available? Okay. Dr. Aarti Chauhan not able to join. She had informed me, so I'm skipping the next presentation, number 11. And we can go for number 12 by Nabavita Sen.
Yeah, ma'am. I am there, ma'am. I can
Yeah,
proceed.
ma'am. Yeah, ma'am. Please, ma'am.
Yeah.
I request
Ma'am, my
Abhijit
share
sir,
is visible, ma'am. My screen is visible.
no, ma'am.
Just a second, ma'am. I'm sharing. Ma'am, is it visible?
Yes, ma'am. Now it is visible.
Okay. Can I start, ma'am?
Yes, ma'am. Yes.
Yeah. Good evening, everyone. This is Navmita Sen, a research scholar from Mangalatil University, Aligarh. My work is mainly on the formulation, optimization, and evaluation of transitosomal gel for the vesicular delivery of the BCS class 2 drug for the anti-inflammatory drug. So here, like whatever I have done in the inflammation, like what is that and how can we defense against the potential harmful substances, and how can we formulate one vesicular drug, that I am going to discuss here. With a key advancement for this type of delivery, like to reduce the oral toxicity for the tablets or any herbal products what we are taking into consideration. So there are the three key advantages for vesicular drug delivery system. That is, first is your enhanced bioavailability by taking this type of gel, reduce toxicity and the protection. The core application where my work, what we have done in the topical and transdermal, but we can also work as an ocular delivery and also as a part of an oncology. That is also very, very important parameters for that type of core applications. Here, as the formulation of gel I have made with the BCS class 2 drugs, here the challenge is to fade the stratum corneum and to go up to the dermal layer for the epidermis part for the topical administration of the drug. Like the BCS class 2 drug, they are mainly, they can't penetrate. They can't penetrate to the your deeper layer for the skin. So by the formulation of the transethosomes, in that what we are making with the edge activators, we are promoting a drug to the stratum corneum, to the epidermis, from the dermis and to the blood vessels. So this is one of the challenging parameter for the dermal study, that whether our drug is reachable to the blood vessels or not. So my formulations is transethosomes I made with the BCS class 2 drug and also the herbal drugs for my project students. So here they are modified version of a traditional ethosomes, what we are incorporating like a liposomes or the your neosomes. So here we are introduction with the ethanol, the phospholipid, and also the component like the edge activator we are adding with the twin 80 to enhance your permeation from the skin. That is a very challenging, whether the drug is going under the skin or not, to have its potential therapeutic activity. Next, this is our one structure where we can see that these are the lipid bilayer or edge activator or ethanol, hydrophilic and hydrophobic drug. What is the main good parameters for the transethosomes is that we can encapsulate both your hydrophilic drug as well as the hydrophobic drugs with this type of formulations. So my slide is visible, ma'am?
Yes, ma'am. Yes, ma'am. It is visible.
Right, ma'am. So here we have made a transethosomes with a BCS class 2 drug, as you see here, and it encapsulator of the hydrophilic or hydrophobic drug is possible with this lipid bilayer for the enhanced activity for the skin. As I am working on the topical drug delivery, where we can do in consideration with the cosmetic products, also we can do with the herbal products. And like the four things what actually we are doing in the lab, one is cold method, hot method, thin film hydration technique, and ethanol injection method. So whether our drug is thermolabile, whether if it is sensitive to the your temperature, then we have to go for the cold method of formulation. And here we are having magnetic stirrer. With this, the drug has to be encapsulated, and with the help of that we are promoting a drug and keep it into the refrigerator for the formulation. And after that, our characterization parameter, that is our evaluation, which is very, very important for the nanotechnology. As transethosomes means we are going for the nano formulation here. So that formulation means with the help of a probe sonicator, we need to sonicate that with a multilaminar vesicle into the monolaminar vesicle, and we have to refrigerate it, and we need to get the your stable formulation for this product. Here, the formulations as I made with the BCS class 2 transethosomes, right? So we are having the gelling agent here like carbopol 934, and also the concentration has to be constrained with the 1% or 15%, depending upon our your availability and the gelling property of the particular solution. Here, one of the very interesting thing, like your sustainable development goals, like the 9, we are going for the your industry innovations and technology, right? To infect the BCS class 2 drug or the API, which is having a very adverse effect and contraindication, like your kidney failure or the congestive heart failure for the person, those who are taking the oral tablets. Like as for example, we are having curcumin, like the herbal drugs, which is a BCS class 4 drug now here. So it is having low solubility as well as low permeability. So how to encapsulate this type, because there is no tablet is there for the curcumin, because it is not absorbed by the liver. So with the help of this, and in fact gingiberine and the curcumin also, we can add it for our chemotherapy. Now the research is already started with that type of therapy. So we can feed this API drugs, and we can use certain herbal formulations for the making of this type of formulations. This is your gel, like the base gel what I have formulated, and these are the trans ethosomal suspension, which has to be embedded into this gel to get it viscosity, the consistency, the pH, the spreadability, the drug content, and all. So after all these parameters, we will see, like the pH of the skin is very sensitive, like it is 6.8. So whether by applying this type of gel, whether we are getting any type of dermal toxicity or allergy or any type of skin your reactions. So that is very, very important for any type of topical formulations as per as dermatology is considered. So here next we have actually, that is a long calculation. I did not kept full in the slide because the time is very constant. So here we are having pH constant your measurement with the help of a pH meter, that is 6.2 is coming. And second, viscosity of the gel is appropriate, like 5210, like below 10,000 it should be. Now drug content has to be done with the help of your your centrifuge, and then the spreadability of that particular gel has to be constrained, whether that spreadability is proper or not for this type of formulation. Next, after this, your characterization, we have to go for some acute dermal toxicity test with the OECD guidelines 402 for the dermal toxicity. So 200,000 and 2000 mg per kg body weight we have taken in the ISC forms, and we have checked about that erythema, edema, skin irritation, ulcerations, or other dermal abnormalities at the application site. So here, as I have kept like 3 picture in the labs, that where we have shed the skin and we have applied like 200 and 1000 and 200 mg per kg of this my hydrogel, and there is no your abrasion or the your any type of redness or swelling has been observed. So the formulation is optimized, and we can say that it is having a good pH range. Next, the second is that as I worked in your inflammatory inhibition, so I need to induce the edema in the rats, and again I need to check upon the grouping of the animals, and based upon that, that 5 groups of the animal has been selected, and based upon that gel formulation, what I have made like 0.5% and 1%, and simultaneously I have checked with your marketed formulations here. So next, that pore volume has to be measured with the help of an instrument known as plethysmometer in the lab. So first of all, I need to induce that edema with a carrageenan induced, and the subplantar injection has to be given to the rat in the pore region and 0.1 ml, and subsequently measured that volume within 1, 2, 3, 4, 5, 6 up to interval 8 hours of after giving that carrageenan induced injections. So here, some types of groups I made for the grouping of the animals in vivo study has been done. So group 1, we are containing normal saline, then second one like this. So the formulation, that is the optimized formulation about the optimized hydrogel, is 1% optimized formulation of the BCS class 2 drug. And it is showing that the inhibition in the pore edema will be 57.60% with this type of formulation. So these are the certain chart calculation depending upon that, like 1 hour, 2 hour, 3 to 8 hours. What are the volume that has been recorded with the help of a plethysmometer? And like this, we are recording that your inflammation in the pore. And after that, we are planning after this the histopathology studies has to be performed. So here percentage inhibition, how can we keep like this table, what I showed you, like this, we have to make the formula for the edema volume, and with that the percentage inhibition edema, like C minus T by C into 100 will give you the exact whatever the volume we are having the control sample and the your test sample we are having. So this is very, very important to having that percentage inhibition in the pore volume. Second, the very, very challenging thing in our formulation, that whether the formulation is having a stability or not. So according to the ICH guidelines, we have maintained our that formulation, and we are keeping that formulation into your consideration, whether the entrapment efficiency, whatever we have taken, whether pH, whether any type of viscosity has been reduced, like 0 degree, 25 degree, or 40 degree according to the guidelines. So we saw mainly it is a stable formulation, and after certain types of days, we did not find any type of your issues in this type of formulation. So further, so many things like the histo, I have already planned, I have already sent the sample, and we are waiting for the report. Once it will come, so I'm planning for that that filing of one of my your anti-inflammatory gel. These are some of my references. Yeah, ma'am. Hello, ma'am.
Yes, ma'am. Yes, ma'am.
Yeah, ma'am. I have finished. Any questions from your side?
Yeah. I request Honorable Chairperson, sir, is there any questions to the presenter?
Ma'am.
Hello, ma'am.
Yes, ma'am.
Yeah, any questions from your side, madam?
Yeah, that's what I'm asking, ma'am.
Yeah.
I request anyone from the participant side or chairperson, if any questions to presenter, please. Okay, there are no questions, ma'am. Thank you so much for your wonderful presentation.
Thank you, ma'am. Thank you so much.
I request Dr. Sujit Jadhav, sir. As of now, we had concluded this technical session 2 also with this presentation, sir.
Yes,
So over to you, sir, for conclusion remarks of day 1, sir.
Yes, I request Professor Maruti Petkar, sir, if he's there, to have some words as a chairperson for this session. Petkar, sir. I think he lost his range.
Hello.
Hello, if you are there, sir, please conclude the session by having sharing some few words regarding this session.
Okay, sir. Okay. First of all, I thanks to all the organizing committee, sir, for giving me a chance working as a chairperson for this technical session 2. In this session, the very nice presentation was given by a different resource persons. In the first presentation, the importance, formulation, and characterization of kojic acids, which we know that it has very huge importance in the field of pharmaceuticals and cosmetic industry. In the second presentation, which was also very nice presentations for the investigation of skin-related diseases, as we know that many of the people are not aware
Best practice आहे का नाही हे असंच
but
द्यायचं
of the
टेबल असंच द्यायचं
or the investigation.
स्क्रीनशॉट काढता येणार नाही
And third one, we also thankful to the third and very nice presentation, which was about related to the investigation of new techniques for the drugs delivery, which was also nice. And I thanks to all the organizing committee for organizing such a nice conference. Thank you, sir. I will hand over to Dr. Sujit, sir. Hello, sir.
Okay, thank you very much, sir. for your presence, and as I have invited you and you accepted our invitation. We are very much thankful to you on behalf of Sidvi Foundation as well as Fulton Education Society, Srimant Bhaiya Sahib Rajamani Mahavidyalaya Maswad. I am very much thankful to you. Very
you, sir.
soon we will send the thanking letter to you.
Okay, sir. Thank you, sir. Thank you so much.
Overall, concluding today's session, very nice and fruitful sessions. 2 sessions we have with us with Dr. Rania Lampo ma'am, who has given speech on conservation, as well as in the second session, we have another distinguished speaker, Mr. Chinmay Sawant, a wildlife biologist. Both of these resource persons, they have given a very fruitful and we can say delightful and as well as very encouraging lectures. Both these lectures are very encouraging to us to work furthermore in the conservation of the nature. So what I conclude, what I think regarding this particular conference, as we have our Fulton Education Society, it's completing now 75 years. We are completing in this year, and on behalf of Fulton Education Societies, this Platinum Jubilee ceremony, we have organized this conference on Nature Conservation Day, where we learn to conserve not only the forests but also the grassland ecosystem is also that much important for the biodiversity, as far as biodiversity is concerned and as far as nature is concerned. This is what we have truly understood from the resource persons, as well as our secretary, Honorable Mr. Srimanta Sanjeev Naik Nimbalkar, who have given the inaugural speech, very fruitful and very honestly, he has concluded all the history of our college and our samstha, which is always encouraging us for having such academic goals. As well as I will talk about our principal, Professor Dr. Vikas Kakade sir, who have encouraged us to organize this conference in very little short period, very little time. He has been granted us the permission to organize this conference, and we have been in along with Sidvi Foundation, we are able to arrange such a type of international conference in our college. And whatever the papers they have been presented over here, I think 6 papers have been 6 presentations have been given by different workers from the overall India, and they have given their papers. One from Uttarakhand, then one from Mumbai University, one from Telangana, Hyderabad, one from Lucknow, one from Gujarat. So various parts of India, different parts of India, we have the peoples that have been reached up to us, and they are willing to present their research work in our conference. We are very much thankful to all these people, all these researchers, and all their guides as well. And I request all other participants who have been listed for tomorrow's presentation. I request you all to be prepared with your PowerPoint presentations and a strong internet connection for during your presentations. The schedule has been we have shared on the group. So please be prepared during your time schedule, as the time schedule has been given to you. I'm very much thankful to Sidvi Foundation, Nakela Madam, who is always with us from last 3 years. We are organizing these conferences And I think Nakala madam and myself, we have been in touch with each other right from 2021, right? That is right from the COVID lockdown period we have. Right from these 5 to 6 years, very closely we have been together. We are organizing conferences, and we are having some research publications as well togetherly. So I am very much thankful to such a personality. Due to COVID, I am able to meet with her, and we have been together and hands in hands. And I hope in further years also we will be together for such different activities. So concluding the today's session, with the grant permission of our principal, our secretary, and Nakela madam, I declare that this today's session has been over, and we will meet tomorrow at 10 a.m. sharp on the given Zoom Meet link. Thank you. Thank you very much, all of you.
Thank you so much, sir, for your wonderful concluding remarks. There is a small announcement that there is a small change in program schedule. We need to add some more presentations for tomorrow. So we'll update that and we'll send you by this evening or night, sir. And that will be sent to all the registered participants' mail IDs, and WhatsApp group also will be sending. Thanks a lot for your all cooperation, and what Sujit sir said is absolutely right. God made us to meet to make some societally useful, productive work, like organizing such a wonderful conferences. SPRM is giving a platform to me to meet all of you at this juncture, and I thank my heartfully to respected principal sir, and the administrative people and management people of SPRM college, and especially Dr. Sujit Jadhav sir. Thank you so much, sir. As you said, tomorrow let us meet all by 10 a.m. Thanks a lot, sir.
Let me have one point I have forget. To
Sir.
all the participants, we should have to know that Nakela madam, Dr. Nakela madam has operation cataract operation from last 7 to 8 days. She is unable to have screen time, and still she is here with us. I am very much thankful to her and the devotion she is giving to organize this conference. Thank you very much, madam.
Thank you, sir. Thank you so much. It's all courage and strength given by you and Almighty God. I had not given my 100% efforts, sir. That is what my pain in this. But I must say my heartfelt thanks to my brother, Mr. R.V. Rashekar, sir. Whatever I had instructed him over phone, even over nights also, he had done all these things. Mr. Rashekar sir, thank you so much, sir. Without you, we cannot reach this. Thank you so much, sir.
Thank you.
Thank you, madam.
Thank you, sir. Thank you so much, sir. We shall conclude now, and tomorrow by 10 a.m. We shall meet, sir. Thank you so much, sir.
Thank you.
Sir, officially, can we stop this meeting now, sir?
Yes, please.
Yeah. Thank you, sir. I request Rashekar sir, please end the meeting all.
Okay, madam.